Welcome to our monthly Science Connects webinar. My name is Dawn Graves, Engagement Associate of Events in the Faculty of Science. The presentation, we acknowledge that the University of Alberta is located on Treaty 6 territory and respect the histories, languages, and cultures of First Nations, Métis, Inuit, and all First Peoples of Canada, whose presence continues to enrich our vibrant community. The presentation today, Brain Immune Cells, is about 30 minutes in length, followed by a question period. We will be collecting questions throughout the presentation. Please type your question into the Q&A box, which will appear at the top or bottom of your screen. At the end of the presentation, these questions will be shared with the presenter to address. A video box of the presenter will appear. If it happens to obscure the presentation material, you can move it by clicking and dragging it to another area of your screen. It is now my pleasure to introduce Martina Kalkunis-Ruppel, Dean of the Faculty of Science. Welcome, Martina.
Thank you, Dawn, and welcome everyone to our Science Connects seminar. This is actually the last Science Connect seminar in our pandemic year series. We're going to be continuing the Science Connects webinars next year. We're going to revert back to our Science Talks title, and we've enjoyed meeting with you so much during this year as we've been through the pandemic. And I want to just give you a couple of updates. The update that many of you will be interested in is operations in the Faculty of Science at the University of Alberta. So we are getting ready to welcome students back in the fall. Our research is going back to before the pandemic in terms of capacity. And we're going to be welcoming students face-to-face. Summer classes are ongoing. And so we're right in the middle of fall preparations. And I hope that you feel a sense of optimism like I feel as we see some light at the end of this COVID-19 tunnel. Another update is that beginning on July 1st, the University of Alberta officially goes through its transformation into the University of Alberta for Tomorrow, where we have a new college structure that sees the Faculty of Science, the Faculty of Engineering, and the Faculty of Agriculture, Life, and Environmental Sciences being united in a College of Natural and Applied Sciences. And I will be stepping away from my role as Dean of the Faculty of Science to oversee that new college. So I'm being seconded away from this role temporarily while we build up the new college. So while I'm doing that, we have Acting Dean of Science, Fred West, who will be stepping in to the Dean role. And at our next webinar, you will meet him, and that will be a webinar that happens on Alumni Weekend after the summer is over. So today, let's enjoy our last Science Connects webinar of this academic year. And I am thrilled to be introducing you to our speaker, Matthew McCauley. Matthew received his Bachelor of Science from the University of British Columbia with a major in biochemistry. He was supervised by Professor David Vocadlo during his PhD at Simon Fraser and began studying carbohydrates, in particular their biological roles in a field called glycobiology. This interest was extended during his postdoctoral studies at the Scripps Research Institute, where he worked with Professor James Paulson. And during his postdoc, Matthew developed an expertise in immunology. Matthew's expertise in immunology and carbohydrates converged when he started his laboratory at the University of Alberta in the Faculty of Science in the Department of Chemistry. And he was joining and bolstering the already excellent carbohydrate-based research that was started by the late Ray Lemieux who is a world-renowned carbohydrate, who was a world-renowned carbohydrate chemist and University of Alberta professor. Matthew's supported by a Tier 2 Canada Research Chair in chemical glycoimmunology. He and his research team are tackling important questions related to immunomodulatory carbohydrate binding proteins in the context of neurodegeneration and autoimmunity. I'm really excited to hear Matthew's webinar today. So Matthew, welcome. And we look forward to hearing all about your science.
Okay, thanks, Martina. I'll get my screen sharing. Okay, so first of all, as a faculty member of the Faculty of Science, I do wish Martina the best in her new role. I'm an assistant professor in the Department of Chemistry. I also hold a cross appointment in the Department of Medical Microbiology and Immunology. That's in the Faculty of Medicine and Dentistry. And I hope you get a little bit from this talk how we marry some chemistry, biochemistry, and immunology to study important immune cells in the brain that I'm going to tell you about today. So on this first picture, what this is it's a section of a brain. It's a mouse model that develops Alzheimer's disease. And the yellow are these what are called plaques, one of the key pathological hallmark of Alzheimer's disease. And you can see very strikingly that around them are these lots of these red dots, and these are the immune cells in the brain called microglia that I'm going to tell you about today. So many of you are here today, probably because you're interested in Alzheimer's disease. Most of us, in one form or another, will have some kind of personal connection to Alzheimer's disease throughout our lifetime. And most of us also know that Alzheimer's disease is a terrible disease with a very high societal cost, as well as emotional cost for the family. Alzheimer's disease, of course, is loss of brain function. In the later stages of Alzheimer's disease, the brain is physically shrunk due to the loss of the death of all the neurons. And so if we zoom in sort of in pictorial over on the right here, the way this works is there are two major pathological hallmarks of Alzheimer's disease. The first I want to tell you about is the amyloid plaques. So this is the pathological feature that develops first. So these can appear decades before any physical manifestation of the disease. And this is then the next stage of the disease is eventually the formation of neurofibrillary tangles. Now, this is inside the neurons, and these NFTs, or neurofibrillary tangles, are actually what causes the death of the neurons. But it's the accumulation of these plaques that occurs very early on, and scientists and pharma realized going back 20 or 30 years ago that if we could slow down these plaques, this could be a potential treatment strategy to Alzheimer's disease. So how do these plaques accumulate? What's going on there? So there's this protein in the brain called the amyloid precursor protein. Its function is not important. What I do want to tell you about is that there are two enzymes that are basically like molecular scissors. They're called secretases, and they clip the protein, they cleave it here and here, and it generates a little fragment of a protein called a polypeptide. And this is called amyloid beta. So the amyloid beta has a tendency to self-assemble into oligomers and furthermore into larger assemblies of fibers or fibrils. If this process keeps going on, we get what's called a plaque, or a senile plaque, or a neurodegenerative plaque. Now, I like to think of these plaques as basically like a boiled egg. And this is an irreversible process. So we know that if we boil an egg, we're not going to get it back to the liquid state. And so once these plaques form, it's kind of, it's a little bit game over There's no taking those away. And so where we need to therapeutically intervene is at these earlier stages of plaque accumulation. And indeed, that's what's been attempted so far. So if we look at sort of the options for treating Alzheimer's disease, the options are not great, to be frank. These are the numbers. This is a graph that really illustrates sort of the dire need for new medicines in this area. So these numbers are comparing the percentages of deaths in a certain condition between 2000 and 2010. Now, these numbers are probably even more striking if we compare 2010 to 2020. What we see is that cancers and circulatory diseases, we have the onset of new medicines. And so this is fantastic. This is one of the advents of modern medicine, is that fewer people are dying from these diseases. But we see quite strikingly that with Alzheimer's disease, it's the opposite, really. Not only has there not been new medicines, people are getting older, and this is the result. So it's not for the lack of trying. So pharma and fundamental research in academics have tried very hard in the last 2 decades. And 2 strategies that they've taken to try and slow down these plaques is by either preventing the molecular scissors from acting in the first place, and these would be with inhibitors of the secretases. And if we look just over on the right, we can see that huge amount of money poured into these strategies, and unfortunately, they get halted at certain phases of clinical trials because they're found to be not effective, or there's often side effects. The other approach are antibodies targeting the amyloid beta to try to clean it up. If we look over on the right, again, huge billions of dollars being poured into this, and largely unsuccessful. So it's not for lack of trying, and in fact, almost the opposite. The amount of effort put into this has been huge. Now, this table was accumulated in 2018, and there was one antibody that made it through past and beyond and into a phase 3 clinical trial. And you may have even heard of it because only a month ago it was approved by the FDA. So after 17 years, there's finally a new drug for treating Alzheimer's disease patients. This is highlighted on the cover of Nature magazine. And what's shown here is in red are the neurofibrillary tangles, basically the dying neurons in a brain. So this is someone that was treated with the placebo and someone that was treated with this antibody, the name of which you can see here. I have a hard time pronouncing it. I think it's safe to say that this example that's illustrated here is maybe perhaps not representative of all people in the study. If we dig into this study and look at actually what happened, what's being plotted here is the amount of NFTs over time throughout the course of this study. And we can see, for example, people given the placebo, they're developing the NFTs along this line here, and people given a high dose of this antibody. By the end of the study, they have slightly lower levels of the NFTs. So it was this difference that allowed the FDA to approve the drug. But we can see that the effect is not exactly stopping the disease completely. And it's really safe to say that among fundamental researchers, there's a healthy amount of skepticism for whether this is going to be sort of the golden bullet for treating Alzheimer's disease. However, it's not all bad. Due to the huge amount of effort that's put into this area, a lot of new things have been learned about the brain, and particularly about how to develop the best treatment strategy for targeting Alzheimer's disease. And it's pointed us in the area of immune cells in the brain that I'm going to tell you about now. So the major immune cell in the brain are these cells called the microglia. So literally, glia is a cell in the brain that supports the neurons, and micro meaning just small. Here's 2 pictures, fluorescence images of the brain. This is in red here are axons of neurons, and you can see all in green, all the microglia. So there's a lot of microglia that support the neurons in the brain. This is a fluorescence image from my lab. This is a mouse brain. Each of the little red dots, very small because the brain is relatively quite large, is each one of those little red dots is a microglia. So you can see that there's a lot of immune cells in the brain. So what do these microglia do? Well, under healthy conditions, they play lots of important roles. Our neurons are constantly dying. That's just a natural fact of aging. And so those neurons need to be taken care of and disposed of appropriately. And then, of course, you have pathogens that try to invade the brain. And the immune cells in the brain protect us from that. Now, under disease such as Alzheimer's disease, when you have plaques accumulating, the roles of microglia become even more pronounced. And studies over the last 5 or 10 years have shown that the microglia are really, really important in that how they respond to these plaques and how they influence how these plaques accumulate in the first place is really, really important. So one of the roles that microglia play in both health and disease is this process I'm going to tell you about called phagocytosis. So literally, phagocytosis means cell eating. Here's a cartoon diagram of a microglia physically engulfing another cell. And so that is the process of cell eating or phagocytosis. But phagocytosis can also be manifest in different forms, not eating an entire cell, but bits of cell or debris. And some of you are probably thinking, well, I know where he's going with this. They could also dispose of the plaques that are accumulating. And that's exactly right, is that they can influence plaque accumulation. So before I get into that further, I just wanted to highlight, many of you are keenly interested in how University of Alberta is doing. And University of Alberta has become this really great hub for microglia experts. So we have 2 senior investigators, Chris Power and Simonetta Scipione. And then these 4 individuals on the bottom are all junior investigators who started their lab about within a year of myself. So we have 5 young investigators investigating microglia, all for different reasons, as you see, and each one has their own specialty. But the combined expertise of microglia is really outstanding here, and my lab has benefited from these a lot. And we're working a lot with Jason and Anastasia. I'll highlight them a little bit later. So back to microglia and how they can influence plaque accumulation. I mentioned earlier that the plaques are like a boiled egg. It's essentially an irreversible process. The microglia can try to eat away at them, but that's a tall task, and they're not able to do that. So where they can influence plaque accumulation is at the earlier stages in eliminating the smaller debris. And so the way I like to think about this is it's basically someone picking up the trash and preventing trash from accumulating on the side of the road, for example. But microglia can play even more intricate roles than just simply cleaning up the trash. At later stages of disease, the amount of plaques become really overwhelming. And so the microglia can sort of mold the plaques to their will. This is the same image that I showed on the first slide. And you see that the microglia are intimately sort of connected with the plaques. And what's been learned over the last, I would say, 3 or 4 years is microglia have this really good ability to pack the plaques into a tight, compact version. And this is really good. This sort of contains the plaques so that they are less likely to interact with the neurons and drive neurofibrillary tangle aggregation inside the neurons that kill the neurons ultimately. Now, if microglia are not able to do this, perhaps due to a bad genetic makeup or environmental factors, plaques develop more of this, what's called filamentous or amyloid form of plaques, and these are better able to kill the neurons. And so the simple view I have of this is that of a garbage dump. You know, garbage dumps in society are gross and disgusting, but they do serve a purpose, of course, because when garbage gets into our environment, such as plastics in the ocean. They generate microplastics, which can be incredibly toxic. So sort of that same line of thinking can also apply in the brain. So before I go and tell you a little bit more about the microglia, let me take a step back and tell you at a high level about how immune cells, such as microglia, are regulated. So here's the really simplified view of things. Immune cells are governed by an accelerator and a brake pedal. Now, in immunology terms, we call the accelerator an activatory receptor. So this would be a protein that recognizes something, for example, something from a pathogen. So an example would be something that's unique to a bacteria or a virus, and it causes the immune cell to be activated and induce a response that can ultimately clear that pathogen. But equally important is immune cells have to have a brake. And these are in the form of inhibitory receptors. These typically recognize things on our own body and prevent immune cells from attacking ourselves, which of course would ultimately develop into autoimmunity. So this is the simplified view, a brake and accelerator, but really it's a lot more complicated than that. That there's many brakes and many accelerators, and the combination of which is instructing the immune cell to mount in a defense or stay silent. Now, skewing the balance between accelerator and gas pedal and brake pedal can be really therapeutically profound. So an example of this is 2018 Nobel Prize in Cancer. And so what these investigators had been working on for 20 or 30 years prior to that is in the area of cancer. Immune cells have a really profound ability to recognize tumors. It's not that immune cells can't recognize and kill tumors. It's that tumors become really sneaky and start to put on proteins that engage the brakes and effectively breaking or halting the ability of immune cells to kill the tumor. And what these investigators did was develop therapeutic strategies to block the interaction, block the brake, and basically that's like driving around in a car with your brake pedal on and the accelerator on at the same time. And what happens when you take the brake off? Well, you accelerate. So I think there's, with the huge success of this in cancer, there's also the thought, and what excites me is whether we could do the same thing in the brain to unlock the potential of microglia to clean up neurodegenerative plaques. Let me just go back here. So in cancer, these investigators knew that this particular brake called PD-1 would be a really good one, a good inhibitory receptor to target. But we didn't really know which inhibitory receptor would be good on microglia. But we can get glimpses of what would be good through these genome-wide association studies, which I'm going to walk you through. So over on the left here is a timeline of DNA sequencing. So first in the early 1950s, when the structure of DNA was first elucidated by Watson and Crick. And then the landmark 2001, where the first sequence of the human genome was released. This took about a decade, something like $100 billion. That's not what I want to highlight. The most important date on this timeline is actually 2006 and 2007. And that's because this is when next generation sequencing was developed. So it allowed investigators to sequence the entire genome of someone in an order of days to weeks, as opposed to a decade, and for a small fraction of the cost. So shortly thereafter, huge studies started occurring in which they would take patients such as Alzheimer's disease patients and healthy elderly controls showing no signs of dementia, and sequencing their DNA and asking, what are differences in genes between these two populations, and what can that tell us about how Alzheimer's disease develops and potential therapeutic strategies. So very powerful approach. These are all the genes that have emerged from these studies in Alzheimer's disease over the last decade. And they're mapped onto a grid that connects them to how the proteins that they encode are functionally connected. Now, what the most profound observation that's came out of all these genes that are involved in Alzheimer's disease susceptibility is that they are important for immune cells in the brain. I think a lot of people early on would have assumed, hey, it's the neurons that are dying. Naturally, we're going to find proteins in the neurons that are important in determining whether someone develops Alzheimer's disease or not. But no, it's actually proteins that are important in immune cells in the brain, the microglia. So if we sort of zoom in on this gene network, we see that right smack in the middle, connected really to has the most connections, is this protein called CD33. And that's what I'm going to tell you about for the rest of this talk. This is the gene, this is the protein that we study. CD33 is this carbohydrate binding protein. And it belongs to a family of proteins that my lab studies. So this protein called CD33 contains two important things. So first of all, it sits on the surface of a microglia. So here's the plasma membrane, and this is the outside of the cell, and this is the inside of the cell. Now, the inside of the cell has the molecular machinery to act as a brake pedal. So it's one of these inhibitory receptors. And on the outside of the cell, it binds to carbohydrates. And so this is where my specialty in carbohydrates comes in and was nicely introduced by Matina, a real strength of the chemistry department for the last really 50 years. So most of you hear carbohydrates and naturally think, what are carbohydrates that we eat doing in the brain? This doesn't really make sense. And it's not carbohydrates that we eat. It's the carbohydrates that are an important biomolecule in our cells, as important as proteins, nucleic acids, and lipids, one of the four basic biomolecules that make up cells. So where are carbohydrates found? They're found on the surface of all cells. So this alludes to the question we put in the poll. So on the top are two examples of mammalian cells, basically our own cells, and these are electron micrographs stained in a way that we can see the sugars, the carbohydrates, and you can see the carbohydrates on the surface of the cell in this fuzzy layer. If we look at a bacterium, it has this same fuzzy layer. Even if we look at viruses, they also have carbohydrates that coat them that are derived from our own cells. And in fact, SARS-CoV-2, of course, cause of the pandemic, is covered with a shell of carbohydrates. So where are these carbohydrates really important? Well, if you look at when two cells bump into each other, what's naturally going to be at the forefront of these interactions is going to be the carbohydrates, as really nicely illustrated in this picture. So immune cells naturally have a lot of receptors that bind to carbohydrates because when they bump into something, they must decide, am I going to attack it or am I going to stay silent? So immune cells have a lot of receptors that recognize carbohydrates that are found in bacteria, for example. These are going to be activatory receptors, induce an immune response. And likewise, immune cells also have a lot of receptors that recognize the carbohydrates on our own cells. These would be inhibitory receptors. And that's where CD33 fits in. It's one of these inhibitory receptors that recognize the carbohydrates on our own bodies. So back to the connection between CD33 and Alzheimer's disease susceptibility. So what these GWAS, the genome-wide association studies, found is that 90% of the population has CD33 that basically looks like CD33 I already told you about. So it's a long version, and since it's in most people, it's the major version, so capital M. That's sort of the nomenclature we're going on here. Now, 10% of people, and by the way, I'm going to color code this for the rest of the talk, that this long version we're going to put in as red. Now, 10% of the people in the population are lucky because they have a version that's smaller than smaller version of CD33, and it's because it's in a smaller percentage of the population. We call it the minor variant or the minor version. So that's how I'm going to refer to it, and I'm going to color code that blue. So specifically what these large-scale studies found is that 10% of these people that that make this shorter version have a decreased chance of developing Alzheimer's disease. Now, it doesn't mean that they have no chance. There's many other genetic factors that play into this, as well as, of course, environmental factors. So one of the major objectives in my lab is to simply understand how these two different versions function. Why does this one cause people, give people a more chance of getting Alzheimer's disease? And why does this protect people from getting Alzheimer's disease? So these are three questions that we're asking ourselves about this role for CD33 Alzheimer's disease. I'm going to read these out, and then I'm going to go through just a few pieces of data that have been collected in my lab over the last 2 or 3 years that helps us understand these, and then I'll wrap up. So the first question is, can we study the role of CD33 in Alzheimer's disease susceptibility within a mouse model? So you might be wondering, what on earth? Why is he bringing mice into this? And don't worry, I'll get to that. The second question is, what is this short version doing in microglia? Up until we published a paper a couple months ago, we really didn't have a clue what it was doing. And the third question is, since this long version, the major version is made in 90% of people, this would be a great target. And if we could leverage and target this version to control microglia in a beneficial way, it could be pretty profound. So we have some ongoing efforts there as well. So let me start with the first question about can we study the role of CD33 in Alzheimer's susceptibility within a mouse? So most models of Alzheimer's disease are very important. And that's because Alzheimer's disease is this really slow progressing disease. We can't simply take the cells out and study them in a petri dish. This is not a good representation of the disease. And so mouse models have been sort of the key to helping us understand better the role of microglia and ultimately how Alzheimer's develops, not to mention testing potential therapeutic strategies. So being an expert in this family of proteins that bind carbohydrates, when I started my lab at the University of Alberta, I noticed that people had been starting to work in mice and try to understand this, but there was a really interesting aspect, a difference between CD33 found in our bodies and CD33 found in mice. And that is that the CD33 in mice is lacking the brake. It's lacking the molecular machinery to act as a brake. And so I thought that's pretty interesting. Perhaps it's not playing the same role as CD33 in our bodies. And therefore, a mouse model might not be appropriate for studying CD33. So to address this, we used some, last fall, the Nobel Prize was for CRISPR-Cas9 technology, and we used that technology to delete the gene for mouse CD33 in mouse microglia. And so that's what we did. And then we took mouse microglia that either have mouse CD33 or don't have it and subjected them to a phagocytic assay. So this is a microscopy-based phagocytic assay. It's quite simple. The microglia are here, as you can see them. We dump in fluorescent beads and the cells take up the beads, and you can physically see the beads. We can then count the percentage of cells that took up beads as a function of how phagocytic the cells are. And on the bottom here, you can see that the cells lacking mouse CD33 took up the same level of beads. And so if mouse CD33 was a brake, we would have anticipated that taking your foot off the brake would accelerate the immune cells and they would have taken up more beads, but that was not the case. Now, turning our attention to human CD33, we can see that this is exactly what happened. When we take away the brake, in other words, we use CRISPR-Cas9 technology to delete the gene for human CD33 in human microglia, the cells take up more beads. So this is our model of what's going on for human CD33. this long version is sitting on the cell surface and repressing. This is sort of the scientific terminology for a brake pedal, repressing the ability of the cells to take up or phagocytose beta amyloid. And so in other words, this long version is a brake pedal. These results also tell us that studying CD33 in a mouse is not going to tell us very much about human CD33. So in further studies, what we've done is using genetic methods, we've introduced human CD33 into the microglia of mice and studied this in the context of an Alzheimer's disease mouse model. So these are really exciting experiments because they build a connection to what's going on in human disease. So I want to give you a little snapshot of these studies. These are ongoing studies, but they're very exciting, I think. And so this is a mouse brain of mice that don't express human CD33. This is an early time point, and you can see in green some plaques that are starting to accumulate. Now over on the right is a brain that has the human CD33, and you can see that the plaques are really getting out of control early. So we're working hard on this, and this is just a little snapshot. We find that the plaques are accelerated when we introduce human CD33 into them. And so that would be expected. We're applying the brake. They're not able to clean up the plaques as well, and so it accelerates. So the great thing about this is it also demonstrates that we have a mouse model that we could test therapeutic strategies. So what about the second question? What is this short version doing to microglia? And let me walk you through two possibilities. I'll try to keep it simple. The first question is nothing. It's not doing anything. If that was the case, we would call that in scientific terms a loss of function. It doesn't do anything. If that was the case, microglia should be better at phagocytosing simply because they have less of the long version. The other possibility is the opposite. So if it doesn't do anything, well, it has to do something. In scientific terms, we would call that a gain of function. And if that was the case, microglia would potentially be better at phagocytosing because they have more of this. So these might sound similar, but they're actually profoundly different. And like I said, until we published our paper 2 or 3 months ago, we didn't have an answer to this, and nobody had investigated this. So this is what we found, is that when we introduced this short version into microglia, they were better able to take up, they were better able to phagocytose. So this is supporting this gain of function hypothesis. So in graphical terms, here's our model where this short version is enhancing this process of phagocytosis in microglia. And it's doing so through processes that we still don't understand. That's why we put question marks. It's okay. In science, we don't always have all the answers, and it's something that we're investigating still. But the important thing to note is that the short version is basically acting like an accelerator. So the two different versions of CD33 have dramatically different impacts on microglia. One acts as a brake, one acts as an accelerator. And so if we apply that accelerator, we would expect to decrease plaques in the context of an Alzheimer's disease mouse model. And that's exactly what we have found. Again, very exciting results, new results coming out of my lab. Not only are plaques decreased if they have this short version, but the soluble version of amyloid beta. So to take you back to this image, here are the plaques over on the right. These are the soluble versions. In mice that have this short version of CD33, there's less of the soluble. And so this is really, really important because, as I mentioned early on, we need strategies to affect these early stages, and it shows that this short version can do that. So let me finish off with sort of some applying the knowledge that we found towards CD33 to try to control microglia. So this takes me to our role as chemists. And during my postdoctoral studies in San Diego, I helped develop this carbohydrate And I won't go through it in great detail here, but suffice it to say is that we designed this in a way that it binds very tightly, and not only very tightly, but very specifically to CD33. And so we took, in my lab here at University of Alberta, we synthesized this using chemical synthesis, and we decorated it on liposomal nanoparticles. So we use liposomes because they're FDA approved, they're safe, and they're really easy to do chemistry on. And so we've started to target this long version by using liposomes that contain this ligand on the outside. And as a control, to make sure the liposomes don't do anything on their own, we have what are called naked liposomes. You might ask, what are these stars? The stars are fluorophores, fluorescent molecules that we put on so we can track where the liposomes are going. So as an example here, we have a microglia or a microglia-like cell treated with a naked liposome, and we see CD33 in green on the surface of the cell, really nicely dispersed on the surface of the cell. No sign of the liposomes, and that's because the naked liposomes have been washed away before imaging. And then if we overlay that with a nuclear stain in blue, we can see really beautiful that the CD33 is a ring around the cell. Now, conversely, if we treat the cells with these liposomes that target CD33, we see a much different staining pattern. Hopefully you can appreciate that. What's happening here is that the CD33 is being driven inside the cell by the liposomes. We can actually see the liposomes inside the cell, and when we overlay it all, it looks pretty different. And so what's happening here in graphical form is that these CD33 ligand liposomes are targeting CD33, driving them inside the cell. And what I'm not showing you, which is even more exciting, is that they increase phagocytosis. And so this is acting like taking the brake, the foot off the brake. And so it's then accelerating this process of phagocytosis. So I know I've told you a lot. I tried to use some analogies. I'll come back to those analogies now, is that these two different versions of CD33, the long version acts like the brake, the short version acts as the accelerator. So these are really fundamental findings that I think will influence how we potentially target CD33 therapeutically. And I also told you about a therapeutic strategy that we're testing with these liposomes that are effectively increasing or enhancing the ability of microglia to phagocytose. So I'll just finish by thanking a few people. There's four key people that work on this project. Dr. Abhishek Bhattacharjee is here. He's a research associate in my lab. You saw his name throughout on the publications. And then recently we had a postdoc, Gazella, join the lab, and she's been a fantastic addition. I also want to highlight, Gazella's not here. We don't have any pictures with her because of COVID. We have two fantastic undergraduates that I want to mention, Arjun Bains and Gaurav Sidhu, both recent graduates of BioSci and are both off to medical school in the fall. And so I want to thank CIHR and GlycoNet for funding the more basic nature of, fundamental nature of the questions that we're asking, and the Alzheimer's Society of Canada and the Weston Foundation for funding the more applied nature. In other words, question number 3. So I think that's it, and I'll end.
Thank you, Matthew. I'm going to join in on a couple of the questions that have come in, and they're not really questions at all. Both Karen and Helen said, brilliant, thank you. Wow, this is fascinating. And I have to agree. It really, we've heard a lot of different webinars this year, and it's really meaningful, I think, to many of us when we see the questions being answered that we can directly relate to our daily lives and the people that we interact with daily. So this was really wonderful. And you're absolutely right. In science, we don't have all of the answers, but I think everybody that's joining us is grateful for the work that you are doing and the work that your collaborators at the University of Alberta are doing. And it seems like you've just got a wonderful group of scientists in your lab. So, and Russ is saying amazing. Now there's a couple of questions and we have about 15 minutes for questions. And I have one question. I also have lots of questions, but I'm going to ask some of the attendees questions. And one question is, how does this work relate to the work of Dr. Dale Bredesen, who has made significant progress on reversing Alzheimer's disease? Not sure if you can address that.
I won't speak to the what they're doing, but I will say that I think most basic researchers agree that to develop an effective treatment strategy for Alzheimer's disease will require to catch it early. I think reversing, especially from late stage, is going to be extremely challenging. If neurons have died, then that's a pretty difficult thing to overcome. So they're making some. So scientists are making some amazing advances in imaging, particularly MRI-based imaging. You saw that on the cover of Nature I showed you. And being able to do this in people like in their 40s and seeing this, decades before any physical manifestation of the disease in terms of memory loss. And so those advances are going to keep coming and allow us to treat people earlier. I'm quite confident in that.
So Matthew, another question that people are interested in is about early diagnosis. And can you comment on newer tests for diagnosing Alzheimer's disease?
Yeah, in the area of cancer, there's just some really amazing work going on blood tests, where you basically sample the blood and you measure thousands of parameters, and it can tell you if you're going to get cancer or not. And there's some real breakthroughs, I think, that are going to come in the next 5 or 10 years on that. They're trying that in Alzheimer's disease as well. That would ultimately be the best, of course, a blood test. I don't think we're there yet. I haven't seen anything telling us that we're almost there. And so the advances that have been made, like I said, are MRI-based sort of contrast imaging agents in the brain, and those are working pretty well. They're more, it's harder to put a lot of people. It's harder to screen a lot of people with these methods.
Yeah, and that the question was, and actually there's a couple of questions specifically about blood tests. So question from Bill, do microglia play a role in other neurodegenerative diseases like MS, Parkinson's, or Huntington's?
Absolutely, all three of them, they play important roles. And so you saw Chris Power and Jason Flemel in the area of MS. So the process of remyelination in MS, sort of when people have episodes, and then after the episode, the body does have the ability to re-heal and remyelinate. The microglia play a really important role there. Simonetta studies the role of microglia in Huntington, sort of similar aspects to Alzheimer's disease and Parkinson's, I believe, as well, although there's nobody here that studies that specifically.
So a question from Jean, who says, traditionally, Alzheimer's disease is associated with plaques and tangles, which you of course talked about. But then she mentions that some years ago, there was a NUNS study that showed that there was a lack of correlation between Alzheimer's with plaques and tangles. So she's wondering if plaques and tangles are the correct model for predicting Alzheimer's disease. I wonder if you can comment on that, Matthew.
Yep, there's still lots of talk about about whether we have it right. You're absolutely right, and as scientists, we need to question whether we have it right all the time. So there are certainly cases where people develop Alzheimer's disease without getting plaques, and there's also cases where people have lots of plaques but don't develop Alzheimer's disease. So it is really, it's complicated. I think what's pretty clear in either case is that the microglia are playing an important role because they show up on these huge tens of thousands of people, these genetic studies. They show up as really just telling us like black and white that microglia are important in the disease.
So there's lots of questions coming in, Matthew. Some of them are about the environment that you talked about, right? So this is all to some extent influenced by environment. And so a question is asked about, could you talk about and there's a few questions that are asking about the environmental factors that might be important, and especially, for example, like how is this related to alcohol consumption, or is there anything in the environment that we know to be important in the development of Alzheimer's disease, or plaques, or some correlate?
Yeah, so those are very good questions. I don't have all the answers to them. What I will say about those questions is that studying environmental factors in humans in terms of Alzheimer's disease is really, really challenging because there's so many different factors at play, and the genetic makeup is very important. And you saw all those genes that I posted that are connected with Alzheimer's disease. So basically, someone's genetic makeup on how susceptible they are is going to be dictated by the versions of each of those. Maybe there was about 50 or 70 different proteins. And so each person basically on this call has a slightly different makeup. And so even running trials of 100,000 or even 1 million people would not give us a clean answer because of what's called heterogeneity of genetics in the human population. We can turn to animal models, and these tell us important clues, but I won't speak to specific environmental factors because that's not my area of research. I know that there is roles pointing towards alcohol, but like I said, that's not my area of research, so I won't speak to that specifically.
Okay, thank you, Matthew. Another question, is the U of A close to doing research involving volunteers as they are with diabetes research?
Yeah, diabetes, since the transplantation center was set up, I can't remember when that was set up, 60s or 70s, that's been going on a long time. No, I mean, what we're doing, we've got some amazing support, like I said, through several societal grants to help push this liposome strategy, and I'm quite hopeful for it. But we have a long ways to go until we would think of putting anything into humans.
Some questions about the liposomes. Do they cross the blood-brain barrier?
That's a really good question. Yeah, really good question. No, they
That's
don't.
for Steve.
No, they don't. And so we have some strategies that we're using to overcome that, but I think I won't get into the minutia of that.
Okay.
I'll say that's a really good question you have.
Okay, so we have some other questions coming in, and they are from some of our viewers that are wondering about whether or not this talk is going to be available so they can share it with their family members. And it will absolutely be available. And I think some people might have had some connection issues, so we'll make sure that there will be a link available. So Matthew, a question that I have is, it seems that the microglia are really, in sort of the most basic sense, it's really the interaction between the microglia and the plaques, right? And not so much with the tangles. Is that sort of a good way of characterizing this, or is that just one approach is targeting the plaques and another approach is targeting the tangles in terms of ultimately getting to therapeutics? And
Precisely. That's an excellent question, and it very well might be that that's what it'll take. So during my PhD, I was actually working on a strategy to tackle the tangles, and that was taken through phase 1 clinical trials. So yeah, there's this idea that you would have to sort of target both arms in parallel. I think if we could ultimately catch it early enough, then perhaps really targeting the plaques might be enough.
Well, I'm being mindful of everyone's time, and I'm going to wrap this up. I do want to thank you, Matthew, for taking your time today to share this science with us. And I don't know that all of the viewers and attendees know this, but Dawn said it earlier, it's really warm here, and I think Matthew is in a really warm room there. So I want to give him a bit of a break. There are lots of people that are thanking you for your presentation and for sharing some of these results. It really offers hope and opportunity. And we'll look forward to hearing about your discoveries moving into the future and the discoveries of all of your colleagues at the University of Alberta working on some of the other diseases. So thank you, Matthew, from the Faculty of Science and from all of us that have joined here today. Just a reminder, we're taking a break for the summer. I hope everyone has a great break. We will have our next Science Connects / Science Talks webinar actually on Friday, October 1st, which coincides with Alumni Weekend. And so we'll be delighted to welcome you back to campus for Alumni Weekend. Everyone, watch for the post-webinar email that will be following after this seminar. It will outline opportunities for how you can support research and programs like Matthew has talked about today. And it's also a way for you to share your feedback. So for now, I'm going to say goodbye. I wish everyone a great rest of the day. Please take care of yourselves and stay in touch. Thank you, everyone.